Hi everyone, my name is Parmis and I am an undergrad student in Dr. Jensik's lab in the physiology department here at Southern Illinois University of Carbondale. This past 1 year, I did a research on what is the effects of different de novo variants on DEAF1 activity. So what is DEAF1? DEAF1 stands for deformed epidermal autoregulatory factor 1. It is a transcription factor that is highly expressed in the central nervous system and it is developed during early embryonic development. So the purpose of this study was to determine the impact of 3 different patient-identified de novo DEAF1 variants on the transcriptional activity of the DEAF1 protein. In order to do this research, first I used human embryonic kidney cells and I plated them in a 24-well plate. And then I transfected these cells using pcDNA3, DEAF1, V205L, C227Y, and A244T. So in these experiments, pcDNA3 stayed at 1. It stayed the same for all of them. And the 3 different de novo variants that I used were V205L, C227Y, and A244T. And I repeated the same experiment. The first time I used a DEAF1 promoter, and the second time I used the BRSK2 promoter. So after I transfected these cells, within 24 hours later, I replaced the media and then I collected my data by using the luciferase assay reporter. And so over here in this figure, in figure 1, you can see that these 3 de novo variants are found in the DNA binding. And for results, we found out that V205L and C227Y, they both lost their repression activity, which means they both changed how DEAF1 functions, whereas A244T, it did not significantly alter how DEAF1 functions. And so for the other experiment, when I used the BRSK2 promoter, I found out that all of these 3 different variants lost their activation activity, and they all altered how DEAF1 functions. And so for the conclusion, we found out that DEAF1 is capable of repressing its own promoter activity while activating the BRSK2 promoter, and these different de novo variants, such as V205L and C227Y and A244T, they change the activation activity of DEAF1, while only 2 of them, V205L and C227Y, change the repression activity of DEAF1. So any mutation in the DEAF1 gene can potentially result in intellectual disabilities, speech impairment, and behavioral problems in autism spectrum disorder. And that is what our research was about this year. So thank you for listening. And if you have any questions, please let me know. Thank you.